go and get a diagram that shows it stage by stage to show how many nuclei the cyst has. When it X's, how many nuclei does the original trophozoite have, and then how it gives rise to the double nucleated trophozoite that we find in the intestine. Yeah, it's a good question, and I apologize. I just need to go pull up a diagram so I don't mess it up anymore. Okay. But they can have cytokinesis and mitosis that are operating independently. Yeah. Okay. All right. So, what we'd like you guys to do is look at a fecal smear from a person that was infected with Giardia. Now, fecal smears, you guys, are so hard. There's so much junk in there. There's so much poo. And there's more poo than parasites, right? So we could spend an hour just looking at junk in the fecal smear. So what we've done is we've set up a demoscope over here with the fecal smears. And this demoscope has the pointer on one of the trophozoites. There are other trophozoites in the fecal smear, but we've just tried to get one in focus in the tip of the pointer. Now, in addition, since these guys are so little, and even with the demoscope, you're like, oh man, that's my stuff. Usually on the lab practical, we'll have maybe like a photomicrograph that goes along with the fecal smear in the microscope, just to make sure you guys are really seeing the outline of the parasite. Okay. For the lab practical, I will always use the trophozoites because they are the most, they have the most variety of shapes. They're the easiest to tell apart. Okay, so if we were working in a diagnostic lab, we would definitely also be looking for cysts. But for me, you know, so often the cysts of the different organisms, they're round, and that's about it. And it's really hard for me to distinguish between cysts. So that's why on the lab practical, we'll put the trophozoites for you all to identify. Okay, they're the easier of the two forms. Okay, all right. So we've got a demoscope for Giardia. Now that's not to say, you guys, we want to discourage you from looking at the Giardia fecal smears in your micro-B boxes. It's just that, like we said, sometimes you can take a long, long time trying to find a trophozoite. So we wanted you to be able to see one, you know, for sure on the demoscope, and then go ahead and take a look at your fecal smear. It's good to have experience looking at fecal smears and see how challenging it can be to find these parasites. Okay, the next one we'll do, you guys, is another flagellate called Trypanosoma Brucei. You'll see here there's two flagellated parasites that are really important in human health. Trypanosoma Brucei causes African trypanosomiasis. African trypanosomiasis, also called sleeping sickness. And the reason is it invades the central nervous system and can cause a coma-like state. Like people say, you know, my daughter can't wake up. It's like she has sleeping sickness. And it's because of the neurological effects of the parasite invading the central nervous system. So it's often called sleeping sickness. Now, this guy, it's, again, you know, these guys, they're so, you can say beautiful, but they're so deadly. This guy, in fact, I noticed, I think Professor Meyer, you guys may have seen this company that makes these stuffed microbes, okay? So this actually helps a little bit in this case. So these trypanosomes, they are single-celled organisms, but they have this beautiful kind of S shape or slipper shape. They have a flagellum that's attached at one end of the cell, and the flagellum arches back over the cell, and it extends long past the body here. And the portion of the flagellum that arches over the cell itself is attached to the cell with what's called an undulating membrane. So as the flagellum is beating back and forth, the undulating membrane is beating back and forth, and that creates the force that helps the trypanosomes move through our bloodstream, our lymph, for example. So, and they don't have eyelashes, though. That's, I just want you to know that that's a little bit of a creative point there. Okay, so if we were to cartoon these trypanosomes, so this would be the body, and here would be the flagellum, coming back like so, and then the undulating membrane would be here. Okay, now, you know, we said these guys can invade the central nervous system, so this is a very serious infection. So how do we get infected? These have an arthropod vector, and the arthropod vector is a biting, blood-feeding fly called Glossina or tsetse fly. Yeah, tsetse fly, right, yeah. So an important way to prevent people getting African trypanosomiasis is tsetse fly or Glossina fly control. Just like here in Sacramento, we try to control mosquitoes that might infect us with Nile virus. In parts of Africa, controlling Glossina is an important way to help prevent people getting infected with Trypanosoma Brucei. Now, the diagnostic sample that you would want from your patient would be blood, peripheral blood sample. So again, we've got a demo slide set up, you guys, of a peripheral blood smear from somebody. Oh my gosh, they're so heavily infected. It makes your heart hurt. But we've got a peripheral blood smear from a person suffering with African trypanosomiasis, and you'll see the trypanosomes, and they are in the blood smear. They are what we call extracellular. They're not infecting the blood cells. They're swimming between the blood cells. Okay? And like we said, this poor person was heavily infected, so it's really easy to see the trypanosomes. Okay? Your micro B boxes, most of the blood smears are pretty good too. So if you just want to look at your own blood smear, that's fine. Now, a related pathogen, also a trypanosome, is called Trypanosoma Cruzi. And this causes American trypanosomiasis, or Chagas disease, named after the Brazilian doctor who did a lot of work. Dr. Chagas was a Brazilian doctor who did a lot of the investigation to figure out the life cycle. It looks basically the same. Most of our blood smears are horrible, you guys, of the Trypanosoma Cruzi. I think what happened, the blood was left out too long, and the cells and the trypanosomes are lysing. So just so you know, on the lab practical, this is the one that we would put on. But we could still ask you questions about Trypanosoma Cruzi. So Trypanosoma Cruzi, it too has an arthropod vector, and the vector is called, there's several different names. You can call it a triatoma bug. It's actually an insect, or reduvid bug. It's the common name that it's often called a kissing bug, and we'll explain why, or an assassin bug. Yeah, so let's see if we can't figure out why the heck it's called a kissing bug. These guys are nocturnal insects. That means they come out at night when we're sleeping. They're attracted to the carbon dioxide that we exhale while we're sleeping. So they follow that carbon dioxide gradient up to our heads, right? Because that's where most of the CO2 is. Now what they'll do is they have a piercing mouth part, kind of like a hypodermic syringe, like a mosquito. So they puncture our blood vessel, and they start sucking our blood. And as they're sucking our blood, they defecate, they poop. And it's in their feces that the Trypanosoma Cruzi are located, right? So how do the Trypanosoma Cruzi get into our bloodstream? Usually, we're sort of half awake, and we feel something on our face, so we go, ah! And we wipe the feces into the bite wound, and that's how the trypanosomes get in. Isn't that? I mean, we help ourselves get infected. Or another real common way is that the infected bugs will bite near our eye, and so we're like, ah, and we wipe the feces into our eye, and then the eye, the conjunctiva gets infected, and they take off from there. Okay, so these guys, the diagnostic stage is peripheral blood. We can see them moving in the peripheral blood, but what's so bad is they can invade heart muscle, they can invade the smooth muscle of the intestinal tract, the esophagus, and they can cause congestive heart failure in long, chronic cases because they damage the heart muscle. And this would be a horrible way to die, you guys. They cause megacolon and megaesophagus. They destroy the ability of the smooth muscles to contract. So you can't through peristalsis.